Good morning. It's November 18th, and this is your daily brief in science. Here's everything you need to know. A new study spotlights a potential breakthrough in treating resistant lung cancers by tackling a master regulator of drug resistance. Researchers show that overactivity of NRF2 drives chemotherapy resistance in lung squamous cell carcinoma, and disabling NRF2 with gene editing resensitizes tumors to drugs like carboplatin and paclitaxel. Using CRISPR-Cas9, ChristianaCare's Gene Editing Institute targeted NRF2 in lung cancer models, reversing resistance and slowing tumor growth. They also tackled a tumor-specific NRF2 mutation, R34G, restoring sensitivity to standard chemotherapy in cells and improving responses in animal models. Delivery relied on lipid nanoparticles to achieve robust genome edits in both engineered and patient-derived tumors, supporting clinical development feasibility. Notably, disrupting NRF2 in as little as 20% to 40% of tumor cells was sufficient to enhance treatment response, suggesting real-world applicability where full editing is unlikely. The work frames a paradigm shift, using precise gene editing to restore effectiveness of existing therapies rather than creating new drugs. The team emphasizes that this approach could allow lower chemotherapy doses with potentially reduced toxicity and may be applicable to other NRF2-driven resistant tumors. Published in Molecular Therapy Oncology 2025 by Kelly H. Banas et al., the study draws on diverse lung cancer lines and in vivo models to support reproducibility. CORIXR's lead programs in HNSCC and LUSC are advancing toward FDA-IND applications with ChristianaCare's Institute contributing to safe, patient-ready translation within a community-based framework. A large real-world U.S. study is evaluating an AI-driven breast cancer screening workflow, and the early results are notable. The Assure analysis, the biggest real-world examination to date, finds RadNet and DeepHealth's AI workflow increases cancer detection by 21.6% versus state-of-the-art 3D mammography, improves positive predictive value by 15%, and keeps recall rates within ACR guidelines. The benefits hold across populations, including more than 150,000 Black women and a 22.7% boost for dense breasts. EBCD is powered by DeepHealth's Breast Suite and has been rolled out nationwide at RadNet-affiliated centers since 2023 to detect suspicious lesions earlier and improve treatment options. The AI workflow combines an AI-based detection and diagnosis tool with a safeguard review, where at-risk cases receive additional radiologist review to enhance early cancer detection. The release includes cautious language about potential risks and regulatory considerations that could affect realized benefits. Standard forward-looking risk disclosures warn of regulatory, economic, and technological conditions that may influence outcomes. RadNet provides media contact information for further inquiries. Leadership at RadNet emphasizes scale, diversity, and real-world relevance, portraying the program as delivering equitable results across racial, ethnic, and density groups. Authors and affiliations indicate collaboration between DeepHealth and academic partners, with disclosures of corporate involvement and potential conflicts of interest. Dr. Gregory Sorensen cites the real-world community center setting and diverse population as strengths, noting AI improves access to specialist-level care. Looking ahead, the program plans continued outcome reporting and site-level performance summaries at RSNA 2025, with near-term metrics on site adoption and aggregated detection and recall data and longer-term signals on stage shift or survival benefits. Data underpinning the findings are controlled by a supervising institutional review board and are not publicly accessible, as they are part of DeepHealth's intellectual property. The article underscores potential implications for broader access to specialist-level care and the benefits of early cancer detection regardless of location. A new antibody-drug conjugate targeting FZD7 shows promise for treating triple-negative breast cancer by delivering chemotherapy directly to cancer cells while minimizing damage to healthy tissue. In preclinical studies, the antibody-drug conjugate inhibited tumor growth in mouse models with no systemic toxicity and demonstrated efficacy in 3D mouse mammary organoid cultures that mimic tissue organization and the microenvironment. Authors note that while clinical advancement requires more research, targeting tumor-initiating cells via FZD7 could offer a more precise therapeutic path for TNBC and may be adaptable to other cancers with similar biology. The study, published in Proceedings of the National Academy of Sciences, was led by Dennis A. Carson and Carl Willert at UC San Diego School of Medicine, with funding from NIH, the Mary Kay Foundation, and CureBound. The Proceedings of the National Academy of Sciences paper highlights a collaborative translational research effort and reports no competing interests. The study is cited as Wu CCN, De Luna N, Hairston E. et al. FZD7 expression marks mammary tumor-initiating cells. Proceedings of the National Academy of Sciences, 2025, 122, 46 E252 2465 122. Broader implications include applying the antibody-drug conjugate targeting concept to other cancers with similar pathways and tumor-initiating cell populations, expanding impact beyond TNBC. The research underscores tumor-initiating cell biology in TNBC and the potential for precision medicine to improve outcomes by limiting toxicities and improving durability of response. Future work includes safety and efficacy testing in clinical trials, molecular profiling of FZD7 across patient populations, and exploring resistance mechanisms to antibody-drug conjugate therapy. TNBC accounts for about 10 to 15% of all breast cancers, is aggressive and often lacks effective targeted therapies, underscoring the need for new approaches. TNBC tends to grow and spread quickly, with poorer survival rates, disproportionately affecting younger women, Black women, and those with certain genetic mutations. In human TNBC cell lines and mouse-derived organoids, FZD7-expressing cells showed higher sensitivity to the antibody-drug conjugate, supporting potential effectiveness across TNBC contexts. A new study in PNAS analyzes about 10,000 stranded or entangled marine animals to measure how lethal ingested plastics are. It finds seabirds are most affected by hard plastics and rubber, with 6 tiny rubber pieces capable of about 90% mortality in some seabird species. Sea turtles face high risk from soft plastics, where 342 pea-sized pieces could prove lethal with 90% certainty, and marine mammals are especially vulnerable to fishing debris, where 28 small pieces can kill a sperm whale with 90% probability. Lethality thresholds vary by species, size, and plastic type, but the dangerous amounts are far smaller than public perception, underscoring the urgency of cutting plastic inputs to oceans. The study provides data-driven insights to guide global conservation efforts and informs policymakers about risk levels and where to target actions. It highlights the impact of balloons, bags, fishing gear, and other plastics, and calls for reducing production, better waste management, and cleanup to protect biodiversity. Findings could inform wildlife protection strategies and plastic pollution mitigation measures worldwide. Experts describe the study as a rigorous contribution that frames plastic pollution as an existential threat to marine life, while noting limitations, such as not addressing microplastics, larger debris ingestion, or entanglement risks. Authors stress that even small amounts can be lethal, and that the data offer a scientific basis for risk thresholds and targeted policies, with a focus on reducing production and improving collection and recycling. The researchers and external experts emphasize defining lethality thresholds to help policymakers craft regulations aimed at reducing production and enhancing waste management, recycling, and cleanup efforts. Global plastic input to oceans exceeds 11 million metric tons annually, with UN projections suggesting this could triple over the next 2 decades. The study focuses on plastics found in stomachs and does not account for chemical effects or entanglement, implying the true harm may be higher. Nearly half of the studied animals were threatened or endangered. The research did not assess all plastic impacts, concentrating on large plastics greater than 5 millimeters and ingestion, and excluding microplastics, sublethal effects, and entanglement. The study is published in PNAS, with international collaboration from institutions including the University of Tasmania, CSIRO, and Universidade Federal de Alagoas. A soft electronic patch worn on a cloth armband uses stretchable electronics, motion and muscle sensors, a Bluetooth microcontroller, and a flexible battery, powered by a deep learning framework that denoises signals in real time to interpret gestures and send commands to machines. This approach tackles a long-standing problem where gesture-based wearables lose accuracy during movement by delivering noise tolerance across a wide range of disturbances. By marrying stretchable electronics with artificial intelligence, the system reliably recognizes gesture signals in real-world, high-motion environments through real-time denoising of sensor data. The work is a collaboration at UC San Diego among Sheng Xu and Joseph Wang's labs, with Xiangjun Chen and Xianjun Chen as co-first authors and additional contributors, Jiwan Liu, Xiaoxing Gao, and Lu Yin. DARPA funded the project under contract HR 001120C0093, and the study appears in Nature Sensors. Originating from an aim to help military divers control underwater robots, the project addresses motion-induced interference across diverse environments. Tests included scenarios with running and high-frequency vibrations, combined disturbances, and simulated ocean conditions using a Scripps Ocean Atmosphere Research Simulator, showing accurate, low-latency performance. Participants controlled a robotic arm while running and under disruptive motions, with additional validation in simulated ocean scenarios. The research demonstrates low-latency, robust validation across diverse environments and suggests broad applicability beyond underwater or specialized settings. The study emphasizes a first-of-its-kind wearable human-machine interface that can learn from individual users and complex environments, potentially benefiting rehabilitation patients, mobility-impaired individuals, industrial workers, first responders, divers, and consumers seeking stable gesture-based controls. The findings, detailed in Nature Sensors on November 17, 2025, indicate applications from medical rehabilitation to underwater robotics and consumer use, highlighting a new method for noise tolerance in wearable sensors and offering reliable gesture-based controls for daily life in dynamic settings. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.